So in this demonstration video, we're going to look at the F1 Flow Cytometry Analysis course and look particularly at this flow cytometry practical. So what we're going to do in this practical is read in a set of FCS files as a flow set. We're going to compensate and transform that data appropriately based on what we visualize and see, and how we can therefore set it up for suitable gating and separation into cell populations, and then summarize and visualize it all together. So what we're going to note is that the resources that were used to design this course are available in the introductory videos and in this video as well, and are listed here. We're going to use these packages called that come from BioC Manager, which installs Bioconductor. So we're going to install these packages and switch over to RStudio now. So to install Bioconductor, we just run this line 75 here, and if it says to ask to install things, we just let that run. We activate it with the library command, and then we can check the package version as well too. We can also install Flow Workspace from the same package. So we're using BioC Manager to install that. We then activate that and check the package version. We can do the same for the Flowspace data too, and check that that package is working and install that too. And the same for Flowcore and ggCyto as well. So ggCyto links to the above packages for visualization. We're also going to try CytoML and tidyverse too. So tidyverse is for parsing our data, and knitr is for linking, creating outputs from the code we write here. So what we're going to do is examine a 96-well plate with GFP positive cells, where each cell has differing GFP tag DNA, so you get the fraction of GFP cells over time. We're going to use the scale function for data transformation. That's in the ggCyto package, not the Flowcore option for scaling, which also can work. What we're going to do as well is download this file from this website and decompress it into our local folder and make sure that it's in our working directory. So if we check our working directory here, I'm in a Flow Cytometry Analysis folder, so I've put these files on there. I've unzipped them beforehand, and these are FCS files. So if you look at the files in that folder, then, oh, I can't see them there. Anyway, there's a bunch of FCS files, and rather than reading in each one individually as a flow frame, we're going to read them together as a flow set. And we can access individual elements of the flow set using these double square brackets. So GS, with the first file will be accessible in this format down here in the console. And the second one as 2, third one as 3 and 4, and so on. So these double square brackets are accessing a particular subset of that data. So we're going to use the read.flowset command to read in many files simultaneously. So we're going to set our working directory and then read these in as a flowset object. We've got a pattern for the file name here, and we can therefore look at what's in FS, that object. So you can see there's quite a few different things. There can be phenotype data if we want to put it in there. We put the names, and then the data itself. So if we look, we can use this pdata function to look at some of the sample information. So you see these are the first 3 sample names. And we can also look at some of the entries there. If we look at entry 1. And what we can do is assign to the well column inside there this substitution command that extracts the well and from the name and adds a new well column. So if we look at the existing entry for that, it doesn't exist. And now what we're going to use is this gsub command. So basically, extracts that information. We can check it by running that part of the command. So you can see there, we've gone from this file name up here to extraction B10 only for the first instance. So these brackets here, the round brackets here, allow us to select that item from the output that we're taking. So we put that as the well, and then we can check in the p data, we get the more simple row names. Now we're going to adjust the column names for the flow set. So if we check the original column names, line 136, we can see that this is what we have here. So we have FSE, AHW, SSE, AHW, and FITC, Pacific Blue A, and time. So we're going to change FITC A for GFP and Pacific Blue for BFP. So we now see the final versions here. So 3 for FSE, 3 for SSE, and 1 for GFP and 1 for BFP. So what we're going to do next is look at a gating set, which is basically a subset of the data by dividing the cells into subpopulations. So let's try that here. So we're taking a gating set from our flow set. So if you look at the structure of the gating set, you can see there we've got several slots in there. So our central principles behind this are to look at how we can subselect certain gates that are of interest. It could be defined by certain approaches, basically to draw a rectangle around our data, or some sort of polygon or polytope, or an ellipsoid, depending on what we want to do. So we can toggle these different features to subselect the cell population of interest that we want to explore more fully. We can set the singlet gate manually, though there are automatic options there that we might use with gate_singlet. With this manual approach can work if you have the same cells or similar cell lines, often where the velocity settings are consistent. And therefore what we would do there is to manually select it is define the gate, verify that looks okay in a plot, add it to the gating set and recompute the gating set with the stats available. We're also going to use a filter ID to refer to these gates, and we'll create a great gating set with which different gates are applied to the data in sequence, so that you have parent and then child gates. So child gates are the kind of more subset of the parent data set. So let's try that here. So we're looking at polygon gate. Using the filter ID is the singlets, and then we're looking at the range for FSE.A and .H, and the range is given there. So looking at 2IE to the 4, and the same for FSEH, a slightly different range there. So we're defining that gate, and if we look at in G.singlets, you can see that it's a polygon gate, polygonal gate singlets. And then we can plot this visually. So here what we're doing is using GCyto to take in that first samples gating set. So that's why we've got GS with square brackets 1. You can look at that there. And the second sample will be GS2 and GS3 and so on. And then we're drawing plotting FSE-A versus FSE-H. So you can see that A versus H on this diagram here. We're looking at the subset of root. And then we're drawing a hexagon around that and gating around the G singlets. And then we're setting the area to correspond to the instrument limits. So if we toggled the bins here to say 100 and Z, you can see that it kind of readjusts how the resolution of the data that's present. So you might want that to be more highly resolved or have a lower resolution depending what's of interest to us. You can see that's very low. If we turn that up to 1,000, it's going to be very, very fine there. So it's very difficult to see there. So that visually kind of shows that to what extent we've captured this. So we might set it back to 200. So we're going to add to our gating set G singlets. So that's another gating set set on there, and we are recomputing that as well. So next we can show where the filtered signals are. You're looking at the FSC versus SCC plot of the live cells. So you can see that aesthetics parameters are changed here. We're still looking at the first sample. And we can see this plot here. So we have some data that's at the edges, extremes, or maybe there's a high kind of exposure rate there. If it did for the same for the second sample, you can see that it's quite similar, and for the 11th sample, it's quite similar again. So if we look at the next plot for the singlets, you see that's quite similar here, except we don't have a higher range for FSCA. See that it's less. And for the second sample, you can see that we actually get a similar pattern. And for the 11th sample, again, we get a similar pattern. And if we toggle the bins, again, it has the same effect that we go down to 50 bins. It's less clearly resolved. And if you go to 5,000 bins, it's going to be so finely resolved that it's actually going to be hard to see. So there, the points are too fine. So now we're going to plot them all together using facet wrap. So facet wrap will iterate through the different samples, and we'll be able to see the tiles per plot using the well column in facet wraps in facet wrap. So let's try that here. So you can see we're plotting the same thing as we had above in the original plot, FSC A and H for the root with the gate around the singlets. And we're adding in the facet wrap command here for 10 columns of data, and we're adjusting the stats so that maybe it will fit within the plot here. So let's move this over so we can see it a bit better. So you can see we have comparable scales on all the different axes, and same with the x-axis. But it's more difficult to plot. What it is showing is the fraction of cells captured within each polytope or polygon. So that's useful to see, and you see for most samples it's pretty consistent, which is good. So we can go back to our steps here. So we're looking at this for the facet wrap. So now we're going to set the GFP positive gate. We're just looking at GFP only here, and we can use a density plot instead of a hex plot above. So here we have the rectangle gate function. We're looking at filter ID for the GFP positive cells with a range from 1000 to infinity to set the gate. So if you see what is in GGFP, you can see that that has a specific range for that rectangular gate. And then we can set that and use the GOM gate to be drawn. So now when we do this plot with GG-Cyto, we're looking at the x-axis is the level of GFP, and we're looking at the density distribution of that for the first sample. So you can see we have the gate g.gfp, and that's the squeezed red area. And we have our limit set there as well. So if you look at sample 11 in the same plot, you can see that it looks quite similar. And for sample 2, it also looks quite similar as well. So what we can do is add that g.gfp gate to the gating set. And that already exists because I added it already. So we're recomputing it with that. And it's the parent set is live for the gating set. So now we can take similar to our plot above, we can plot do a density plot of the GFP positive cells using defined range using facet wrap for all the different samples we have. So for all these plots, we could write them out with with PNG or PDF or whatever, depending on what we wanted to, how we wanted to print them out. So yeah, sometimes these plots take a minute to appear, and sometimes you have to rerun the command in case something didn't quite activate in time. You can see there for each plot we have the area detected for that particular subset. You can see that for some of them it's quite small, and then for others it's larger. So this is much more variable for across the different samples. So now we can go here and get the population stats for downstream analysis. We're going to get the data for gating set from the gating set, and we're going to use this function gspop get count with meta, which also retrieves the metadata and sample names. So we're going to use this function on the the gating set, just to get the percent of parent in PS. So if we look at PS, you can see there has the all the different samples and then the gating sets that we created. So if singlets live and singlets live GFP positive. So if we look at the percent of parents, we're going to mutate a new column, PC of parent, which is count divided by the parent count. So for what we're going to do then as well, select from this data set the sample name, the well, the population, the count, the parent count, and the percent of parent inside there. So if we look at that distribution of the count over parent count, count divided by PS dollar parent count, you can see our values there, and you get a sense of how they're shaped from this histogram. That's roughly the percentage of parent that we see across our different samples. And we look at it there with a higher resolution. So we see most of them are quite a high level. So we can put certain columns you want of interest into a new D4 column. So you see the original PS one had quite a few different columns, and for D4, just want less of them. So you just want the sample names, the wells, which are the short sample names, the population, the 3 populations per sample, the count we had, the parent count, and the percentage of parent. So if we look at D4, we can see that the for the different groups, the percentage of parent differs considerably. Some have very high rates, and some have very low rates. We can also visualize this data using PCA. So most of the variation we'll see will be organized across different eigen values, PC1, 2, 3, 4, 5, and so on. And we'll use the eigenvectors then to plot the coordinates on a plot to visualize it. So what we can do is take our row names from D4. So you see the row names here are just numbers. So what we're going to do is take in the sample names. So there's the sample name there. The wells for each row, which is B10, the population, which could be just singlets in this case, and we're going to join them all together. So if we take this command, what we're doing is that we're just pasting them all together. So if we look at sample 1, this is what we get at the end. So if all that metadata linked together for that particular sample, indeed, we could actually remove the sample name here. It's not really essential. So if we try that revised version, you can see that we have that inside there, and we create that as the row names and see that these are the revised row names. So we have the relevant metadata built inside there. We can create a new data frame D5 based on that. So we're looking at the count, the parent count, and the percentage of parent in there, as well as the row names in table D5. So it's a more simplified table now. So for PCA, we need numerical data. So we can run this function prcomp to compute that. We can't keep the character data inside a PCA plot, PCA analysis. So that will look for correlations across these 3 different variables: count, parent count, and percentage of parents. Of course, percent of parent is going to be correlated with these because it's based on the previous one, so it's not really relevant here. So it'd be wiser to leave it out. And we look at PC values 1, 2, and 3 here just for illustration. And we're grading my colors, which are a function of the dimensions of our alignment. Oh, sorry. We don't need this here. We're going to use the rainbow colors. So if you look at the rainbow colors, if you look at 3 of them, it just has 3 different colors here that we'll visualize down here. So that's PC 1, 2, 3. So what we're adding up here is the sum of squares for PC eigenvalue 1 divided by the fraction of the total. So that should count for the predominant under variation across the full set. So you can see that is 98.5%. PC2 is 1.5%. PC3 actually has effectively 0. So what we can do is then plot this visually to see how it comes out. So you can see there that most of the variation is across the x-axis. The y-axis contributes very, very little. And that there are some groups of samples that are more tightly clustered with each other, and some that are more distant. If we try PC1 versus PC3, this may not resolve because there's so little variation on PC3, so that's not relevant. And we can also try this package factor extra, which can also be used to visualize PCA plots, but it has additional features, but sometimes it can be quite crowded. So we're using this function f is PCA ind, which takes on board our PCA function from above. It has the specifics on the colors for individuals. We're repelling the text if there was labels present. We set the label size to be 0 here, so the labels are not present. We set the text size to 1, and our grading columns are just going to be an orangey color here. Then we're using the plus sign to add on the legend position, which is going to be absent here, in fact. And then we're adding a title. To visualize this plot, we use plot to actually produce it. And that should show a plot shortly once it's completed. There we go. So you can see there we kind of 3 different groups where there's some labeling being put on there, but it shows sort of pretty much the same sort of thing. So what we have here in, if we look at D4, then as our allocated samples across the different populations that were of interest, with the percentage of parents listed there for different samples. And we can evaluate this further using different approaches depending on what we want to do. And we can talk about that later during the class itself.